Our group chose a paper that is looking at the novel role of HSP90 in regulating osteoclastogenesis by abrogating RAB11B driven transport. We are going to talk about protein and vesicular transport within a cell. Our group chose a paper looking into how RAB11B is required for regulating the vesicular transport of cellular cargoes, of which HSP90 is a vital element. RAB11B is a gene that serves as an important component of the vesicular machinery. It is required for the transfer of internalized transferrin from the recycling compartment to the plasma membrane, of which active RAB11B as well as GTP hydrolysis is necessary. HSP90 is a heat shock protein with a mass of around 90 kilodaltons that is highly abundant and ubiquitous molecular chaperone, which plays an essential role in facilitating ER to Golgi vesicular transport by both promoting vesicle targeting and maintaining the structure of the Golgi.
Osteoclasts are cells that degrade bone to initiate normal bone remodeling and mediate bone loss in pathologic conditions by increasing their reabsorption activity. They start off as hematopoietic stem cells, then differentiate into osteoclast progenitor cells, followed by mononucleated osteoclasts, and then multinucleated osteoclasts. The multinucleated osteoclasts would then be used for bone absorption. Here's a picture of an osteoclast. Highlighted in pink, you can see the cell membrane, and inside the membrane, there's 9 nuclei.
So our paper has 2 primary questions. The first question is addressing what the role of HSP90 and RAB11B mediated inhibition of CFMS and rank receptors is in osteoclasts. While our second question is looking at how does the HSP90 RAB11B mediated vesicular transport stimulate the lysosomal deterioration of osteoclastogenesis. This is a model showing the role of HSP90 in regulating the lysosome-mediated proteolysis of CFMS and rank surface receptors via directly modulating RAB11B function in osteoclast development. Both HSP90 alpha and HSP90 beta, identified as the RAB11B protein effectors, couple with RAB11B to improve the transport of CFMS and rank surface receptors to lysosomes via the axes of early endosome, late endosome lysosomes for the proteolytic degradation. This ends up diminishing the bone resorbing activity of osteoclasts. Now to focus in the receptors. We can see how rank and CFMS are the cell membrane receptors that, when bound to rankle and MCSF, produce the signal cascade that promotes osteoclastogenesis, which is a key topic throughout this paper and describes the fusion of osteoclast precursors to form multinucleated mature osteoclasts.
First, the researchers wanted to find out if HSP interacts with RAB11B. Through immunoprecipitation, isolated RAB11B showed HSP90A and HSP90B bound to it. This could be seen here and here. Immunoprecipitation isolating HSP90A and HSP90B shows that the 2 HSP subunits did not interact with each other. To further investigate the interactions between HSP90 and RAB11B, researchers generated GFP overexpressing RAB11B cells. In IF, when stained with HSP90A and HSP90B, researchers could see co-localization between the 2 proteins, as could seen in the merge photo here. Next, the researchers wanted to validate the GFP-generated cells. Through immunoprecipitation, isolated GFP RAB11B showed binding to HSP90s similar to the wild type.
After seeing that RAB11B and HSP90 alpha and beta did interact, they wanted to see if they interacted at the HSP90 ATPase domain. To do this, they used a protein called 17-AAG, which inhibits the HSP90 ATPase domain. When treated with increasing concentrations of 17-AAG, they saw that the HSP70 pathway was activated. The HSP70 pathway is a different regulatory pathway of osteoclasts, but also interact with RAB11B. Here on the right, you can see the relative densities of these proteins when treated with 17-AAG. You can see that HSP70 increases with increasing concentrations. It's also important to note that the density of RAB11B did not change. Here we can also see how 17-AAG affects the interaction of HSP90 alpha and HSP90 beta with RAB11B. We can see that with increasing concentrations of 17-AAG, the binding affinity with RAB11B decreased significantly. Now to graphically show the data from the previous slide, we have immunoprecipitated HSP90 and RAB11B percentages. With the increasing concentrations of 17-AAG, we see that RAB11B interactions with HSP90 alpha and HSP90 beta decreased. This shows that even though the density of RAB11B protein did not change, the interaction does change. Overall, experiment 2 shows that HSP90 ATPase domain is important in the interactions and processes of HSP90 and RAB11B. And when this interaction is no longer viable, other osteoclast pathways are then activated.
Next, the researchers wanted to investigate osteoclast differentiation by blocking HSP90 and RAB11B interactions. TRAP is a protein that's expressed by osteoclasts. Researchers stained for TRAP and GFP overexpressed RAB11 cells that were treated with siRNA targeting HSP90 subunits. Figure A shows the TRAP staining, and Figure B shows the quantitative analysis of Figure A. Figure B shows that blocking HSP90 subunits showed an increase in TRAP positive cells. Next, the researcher investigated downstream transcription factor expressions for osteocladogenesis and found that when blocking HSP90 and GFP overexpressing RAB11B cells, there was an increase in osteoclastogenesis transcription factors. In addition, nuclear fractioning shows that these transcription factors were present in the nucleus.
This diagram here explains the NFATC-CFOS signaling cascade in pre-osteoclast cells, taken from a paper in 2019. NFATC and CFOS are both transcription factors that promote osteoclastogenesis, and following the rank L binding to rank receptors in pre-osteoclast cells, these signaling cascade is induced that leads to the expression of genes that promote osteoclast differentiation. As seen in experiment 3, these transcription factors are upregulated when the interaction between RAB11B and HSP90 is inhibited. The next experiment takes a look at proteins upstream in the signaling cascade. Specifically, the box proteins, we have IKB, ERK, P38, and one that's not shown in this diagram, AKT, were the focus of the next experiment. So now experiment 4 looks at if suppressing the HSP90 subunit will affect upstream signaling pathways. From our diagram before, we are looking at P38, IKB alpha, AKT, and ERK. When we're looking at the control, which is RAB11B overexpression, we see a decrease in the phosphorylation of all of these proteins. However, when we inhibit HSP90 alpha and beta, we then see that these effects are reversed. This further shows that the effects of RAB11B and HSP90 within pre-osteoclast cells decreases the activity of the signaling cascade that leads to osteoclast differentiation.
This is the last experiment from the paper that we are going to be looking at, and the question they are asking is, does the interaction of RAB11B with HSP90 affect lysosome activity? So to assess this interaction between RAB11B and HSP90 on lysosomal activity, cells were transfected with either si-HSP90 alpha and or si-HSP90 beta. And when you treat using either of these, there is a decrease in rank and CFMS lysosome proteases, which you can see here in this blue outline. And then for figure B, the cells are fractionated into two separate fractions, including the biotinylated fraction and the total fraction, which is referred to as WCL. And the surface and total levels of CFMS and rank were assessed by biotinylation pull down in IB. And so we can see here again in these blue boxes that the biotinylated regions or fractions show increased surface proteins. Finally, here in figure E, this figure is evaluating the rank and CFMS lysosomal protease with treatment of ATPase domain inhibitor. And when treated with this ATPase domain inhibitor, 17-AAG, box in the bottom, less lysosomal proteases are expressed. And in figure F, inhibiting the ATPase activity shows an increase in the surface markers. HSP90 and RAB11B interactions both influence proteolytic activity of rank and CFMS.
In summary, rank and CFMS are important in differentiation of osteoclasts, serving as receptors for RANKL and MCSF. The binding of these ligands stimulates the signaling cascade that leads to osteoclastogenesis and the absorption of bone. The interaction between RAB11B and HSP90 alpha and beta ultimately decreases osteoclastogenesis by mediating the transport of these receptors to lysosomes where they will be degraded. As seen in this diagram, RAB11B and HSP90 alpha and beta are involved in the vesicular transport of these receptors from early to late endosome and eventually to the lysosome. Breakdown of these receptors halts osteoclastogenesis through removing the receptors that stimulate the signaling cascades.
This experiment is important because it allows us to further understand how osteoclastogenesis is regulated through vesicular transport.